Welcome to today's lecture on cell biology, focusing specifically on the fascinating topic of vesicle trafficking. In this lecture, we will explore the intricate process of vesicle budding, the role of coat proteins, the importance of coatomer proteins, the mechanism of vesicle fusion, the various endocytic pathways, intracellular sorting, retrograde transport, exocytosis and secretory pathways, regulated secretion, autophagy, multivesicular bodies, and finally, the significance of vesicle trafficking defects in neurodegenerative diseases. This intricate process ensures proper transportation of specific molecules within the cell. Coated vesicles, like those formed during clathrin-mediated endocytosis, play a crucial role in capturing cargo molecules. Proteins like adaptins and dynamin help in cargo selection, recruitment, and vesicle detachment. Coat proteins play a crucial role in vesicle trafficking by aiding in the formation of vesicles and selecting cargo. Clathrin coats are involved in endocytosis, while Copy and Copy coats participate in Golgi to ER and ER to Golgi transport. Regulation of coat protein recruitment involves factors like membrane composition and cargo molecules. Coatomer proteins, or COPs, are a group of proteins that form the Copy complex. This complex helps transport materials from the Golgi apparatus back to the endoplasmic reticulum. The Copy coat is made up of 7 protein subunits, each with specific roles in vesicle formation and cargo recognition. Coatomer proteins interact with receptors and other molecules to sort and package cargo for retrograde transport. In summary, vesicle fusion is a crucial process that involves the fusion of vesicles with target membranes to transport cargo. The snare complex, comprising V snares on vesicles and T snares on target membranes, plays a key role in membrane fusion. Other regulatory proteins ensure specificity and accuracy. Endocytosis is a process where cells take in substances from the outside. There are 3 main types of endocytic pathways: clathrin-mediated endocytosis, caveoli-mediated endocytosis, and macropinocytosis. Each pathway has its own specific mechanism for internalizing and transporting molecules. Intracellular sorting mechanisms ensure that cargo molecules are correctly sorted and delivered to their intended destinations within the cell. Sorting can occur in different compartments, such as the Golgi apparatus, endosomes, lysosomes, and trans-Golgi network, TGN. Various sorting signals, including protein motifs, glycans, and lipid modifications, guide the sorting process. Sorting signals can be recognized by specific sorting receptors and adapter proteins, such as AP complexes. These proteins help in cargo selection, coat recruitment, and vesicle formation. The sorting process is further regulated by small GT passes, which control the targeting and fusion of transport vesicles to their respective compartments. Retrograde transport is the movement of vesicles and proteins from later compartments back to earlier compartments in the secretory pathway. This process is important for maintaining intracellular organization and function. Copy-coated vesicles mediate retrograde transport from the Golgi to the ER by recognizing specific retrieval signals on cargo molecules. These vesicles deliver the cargo molecules back to the correct location in the ER. Exocytosis is a critical cellular process that involves the secretion of molecules and transport of membrane proteins to the cell surface. It is essential for various physiological functions like neurotransmitter release, hormone secretion, and immune responses. Exocytosis occurs through 2 pathways: constitutive secretion and regulated secretion. Constitutive secretion involves the continuous release of proteins and lipids from the Golgi apparatus to the plasma membrane. These proteins lack specific sorting signals and are transported to the membrane through vesicles originating from the trans-Golgi network, TGN. On the other hand, regulated secretion is triggered by specific stimuli and involves the fusion of secretory vesicles containing cargo molecules with the plasma membrane. Regulated secretion is a tightly regulated process that allows cells to selectively release specific cargo molecules in response to external signals. This mechanism is essential for neurotransmitter release in neurons, hormone secretion in endocrine cells, and immune responses involving cells such as mast cells and eosinophils. Regulated secretion involves the storage of cargo molecules in specialized secretory vesicles, which accumulate within the cell and await appropriate stimuli. Upon stimulation, calcium ions enter the cytosol, triggering a series of events that lead to vesicle fusion with the plasma membrane. This process allows the controlled release of cargo molecules into the extracellular space. Autophagy is an essential cellular process that involves the degradation and recycling of intracellular components. It serves as a quality control mechanism by eliminating damaged organelles, protein aggregates, and invading pathogens. Autophagy occurs through the sequestration of cargo within double-membrane vesicles called autophagosomes. The formation of autophagosomes is a multi-step process that involves the coalescence of membranes from the endoplasmic reticulum, Golgi apparatus, and plasma membrane. Several autophagy-related, ATG, proteins coordinate the assembly of the autophagosomal membrane and the subsequent fusion with lysosomes, leading to cargo degradation. Multivesicular bodies, MVBs, are endosomal compartments that contain intraluminal vesicles, ILVs, formed by inward budding of the endosomal membrane. ILVs are generated by the action of the ESCRT, endosomal sorting complex required for transport, machinery, which consists of several protein complexes, including ESCRT-I, ESCRT-II, ESCRT-III, and the associated AAA-ATPase, VPS4. MVBs represent an important intermediate in the endocytic pathway, facilitating the sorting of cargo molecules into ILVs. Furthermore, MVBs can fuse with lysosomes, leading to cargo degradation, or participate in vesicle trafficking processes to deliver cargo to other compartments, such as the plasma membrane. Vesicle trafficking defects have been implicated in various neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, and Huntington's disease. These disorders are characterized by the accumulation of intracellular protein aggregates and impaired cellular functions. Mutations in genes encoding proteins involved in vesicle trafficking pathways can disrupt normal transport processes, leading to the mislocalization and aggregation of proteins involved in disease pathology. For example, defects in the SNARE complex components have been associated with compromised neurotransmitter release and synaptic dysfunction in Alzheimer's and Parkinson's diseases. To sum up, vesicle trafficking is an intricate process that plays a vital role in cellular transport and communication. It involves several steps and pathways, impacting neurodegenerative diseases. Studying this process enhances our understanding of cell function and offers potential therapeutic avenues for various disorders.